right, Tyson, take it away.
Okay, well, welcome everybody. Welcome back if you were with us yesterday. Today we're having our ecology seminar with Dr. Ivette Perfecto. She was born in Puerto Rico. She went to University of Michigan, which is where she got her PhD, where she teaches currently. She's done a lot of this agroecology type work. She's worked around the globe with lots of people, very collaborative, done a lot of work in Mexico and Puerto Rico, which is, I'm sure, what we'll hear of today. She's also written chapters within textbooks, written her own books, published many papers. She's very fun to listen to yesterday, and looking forward to listening to her again today. So when you're ready, Yvette, go ahead.
Great, thanks, Tyson. Let me put my presentation. Okay, so the title of my presentation is Using Natural History to Inform the Study of Complex Ecological Systems, a case study of the coffee agroecosystem. And I put a slide here, a video of the coffee farm where we work, just to give you a sense of what it looks like. It's a very fun system to work with. Thank you very much for inviting me. It's a pleasure for me to be here today, to be with you, and thank you, all of you, for coming to this presentation. So a lot of my work uses natural history and complexity science to try to understand the ecology of agroecosystems in ways that can help us manage these systems more sustainably. My two books with my colleague and partner, John Vandermeer, that are called Ecological Complexity and Agroecology, and the other one is Coffee Agroecology, highlight this approach that we take in our research. Now, before I get started, I want to thank and recognize the many people that have made this research possible. And that includes the many graduate students that have participated in this research from my lab and John's lab. And also our field assistants and coordinators, both in Puerto Rico and in Mexico. And of course, the farmers who have welcomed us to work on their farm. More than 850 species of insects are known to feed on coffee. And of these, about 200, a little bit more than that, are known from the neotropics. However, in spite of that, very few are reported as real pests in coffee. For the last 20 years, our lab has been working on the ecology of the coffee system, focusing on herbivores, pathogens, and their natural enemies. The coffee system is a great model system to study ecological interactions because it's a managed system, so there's a lot of manipulation you can do with it. It's also a perennial system, so the insects are there all the time, the plants are there all the time, and it is a managed system that is managed in a variety of ways. You probably have heard of the shade and the sun coffee systems. Those of you who have heard anything about coffee production probably have heard about shade coffee and sun coffee, which are the two extremes of the management system. And you can see them here, these two pictures. But the reality is that rather than this binary classification, coffee production can be organized along a gradient. Or can be, think of it along a gradient of intensification that goes from these very, very diverse systems that are in Mexico, they call them rustic systems or mountain coffee. And this is basically the original forest, but the understory have been modified and people have planted coffee in the understory. So it maintains a lot of the diversity of the natural forest. Then you have this traditional polyculture, what Moguel and Toledo call traditional polyculture, which are still very diverse. And the system where we work is one of these traditional polycultures, very diverse, a lot of shade trees. and a relatively high density of shade trees. To the commercial polyculture, which has less diversity and less density of trees, but still some diversity there, usually fruit trees and things like that, to the what is called the shaded monocultures, which are systems like these that have shade trees, but they're not not in very high densities, and also is usually one single species of shade tree. In many cases, a nitrogen fixing tree. This is a legume here, Inga, but it could be, you know, a number of many different nitrogen fixing trees. And finally, the sun coffee, or the unshaded monoculture, where it's just a monoculture of coffee. So you have this gradient, no? And our lab have been studying how, as coffee farms are intensified, associated biodiversity declines dramatically. No, you lose a lot of the wildlife in this system, the wild biodiversity. And with the loss of the biodiversity, you also have the loss of these intricate networks of interactions that we have documented result in what we call autonomous pest control. So here we have part of the interactions that we have uncovered studying the coffee system, and these are the four main coffee pests. When you eliminate the shade trees, you eliminate this ant because it's an arboreal ant. And with the elimination of that ant, the whole network breaks down essentially and can lead to this is the monoculture here. When you eliminate those shade trees, then you eliminate the ant. No, the ant doesn't have a place to nest, and that can lead to the outbreak of these four pests. Now, in this talk, I want to focus on the green coffee scale, Cocos viridis, which you see here in this picture. This is a hemipteran herbivore that can reach very high densities in some farms and under some conditions. It is a pantropical insect and can attack many different species of plants. It's not a specialist in coffee. You probably, if you have a lot of house plants, you probably have seen this insect because it's very common among, you know, house plants and greenhouses and things like that. So what I want to do today is give you 3 vignettes about related to the work that our lab is doing. And how we use natural history and complexity theory to try to understand this particular pest. And then if I have time, I'll talk a little bit about the ecological games, one ecological game that we developed that is based on these interactions. Hopefully I'll have time toward the end. So the first vignette is related to ant mutualism and the Turing process. And I'll explain what that is. Then the second one is about the beetle predator and critical transition and hysteresis. And then the third one is fungal pathogen, chaos, and hysteresis. And then, as I mentioned before, if I have time, I'll talk about the ecological game, Azteca chess. Okay, so here's Azteca sericiazur. This is this ant, I'm going to call it Azteca from now on, is tending scale insects. So the scale that I mentioned, Cocos viridis. This is a typical ant hemipteran mutualism where the ant uses honeydew that is produced by the scale. And the ant protects the scale against its natural enemies. This is a very important relationship for the survival of the scale, but it also adds an important spatial component to the system. So let's look at the natural history of the ant and how that spatial component, that spatial pattern formation happens. So to study this ant, which is an important predator of other coffee pests in the system, and as I mentioned, has this mutualistic interaction with the scale insect, we established a 45-hectare plot in a shaded organic coffee farm in Chiapas, Mexico, where we work. In this plot, we geolocated all the trees that were bigger than 10 centimeters in circumference. And it's a big plot. 45 hectares is fairly big. This is, I'm here in one of the regions of the farm, and you can see all the way to this ridge here that has those trees. The plot gets almost to that area, so it's actually a pretty big area. So we geolocated all the trees. Here is a map of all the trees. All these little dots here are trees that are bigger than 10 centimeters in circumference. We basically, in the first time that we did this, by the way, we do this census every year. And the first time we did the census, there were 11,000 trees on the plot. At some point in time, the farmer decided to intensify a little bit the farm, or open up the canopy a little bit, and he cut some trees, or they cut some trees, and we were left with 7,000. But then now we're back to 11,000. The trees grew back, and we're back to 11,000. And we identified 90 different species of trees, so it's fairly diverse. And so one thing that we did after having the map of the trees is locate all the trees that had the Azteca ant nesting on them or in them. And one thing that was clear from this was that the ants were distributed in a cluster pattern, as you can see in this map here, no? And the colors, forget about the colors, they just mean this is a cluster, this is a cluster, different colors, different clusters, no? Initially, we thought that the cluster pattern could have an exogenous cause. That is that this ant spatial distribution was determined by some external factors, such as the level of shade or density of the trees, or particular species that the ants prefer to nest on. And if these particular species were distributed in a cluster fashion, then the ants will be distributed in a cluster fashion. We tested this hypothesis and found no evidence whatsoever for an exogenous pattern formation, which led us then to explore the possibility that this was a self-organized pattern. That is, that this pattern emerged from the internal dynamics of the ant itself and its ecology. No? So we also discovered some other aspects of the natural history of the ant, and that gave us some clues as to what the pattern, how this pattern is formed. So the ant nests arboreally, as I mentioned before, and when the colony gets to be really big, it starts budding out. And it goes basically, it has multiple queens. So a group of workers with a queen will move to another tree farther away and establish a new nest in another tree. And so it would be easy to see how these ants start expanding from one single tree onto a cluster of trees. But if this is the only process that's going on, then you will expect that eventually the entire farm will be filled with these ants. And that's not what we see. So there must be something that is suppressing the ant and limiting its expansion. And that suppressor turns out to be a fly. And this is a parasitoid, a fly parasitoid in the family Phoridae, and the genus Pseudactyon. These are very common parasites of ants. And in this case, the fly oviposit in the base of the head of the ant, and then the larvae gets into the head capsule, the head capsule, and start eating the inside of the head capsule of the ant, and then pupates. And when it's ready for the fly to come out, the head of the ant falls off, and the fly comes out, as you can see from this wonderful picture from Sanford Porter, who works a lot with these phorid parasitoids. Here you can see the head of the ant and the fly coming out. Another interesting aspect of this system is that the main effect of the fly that the fly has on the ants is a trait-mediated effect because it changes the behavior of the ant. Basically what happens is that when the flies arrive, the ant, to protect themselves, reduce their foraging activity by about 50%. And this is a comparison to the work from my colleague Stacy Philpott, a comparison on areas with and without the phorid. Within the same farm, you can find phorids in some areas and not in others. And so you can see that the reduction in the activity of the ant when the phorids are present. And so when the phorids arrive, the ants reduce their foraging activity, and that reduces the rate of acquiring the honeydew. And it reduces the protection also of the ants on the scales. So the cluster pattern of the ant is formed in a process similar to that described by Alan Turing in his famous paper published in 1952, The Chemical Basis of Morphogenesis. I don't know how many of you have read this paper. It's a fun paper to read. Turing was interested in the chemicals involved in the creation of pattern, spatial pattern during biological development or morphogenesis. In chemistry, a reaction is frequently assumed to be stabilized by the balance between an activator or an activation process and a repression process. Now, a different stabilizing process is diffusion, no? So here you have that activation and repression. Here you have diffusion. And so you put a drop of a dye and it diffuses, no? And it's a very stable process. Now, this is also sometimes called well, actually, before I say that, in chemistry, when you well, we should assume, it makes sense to assume, let's say, that if you put these two processes together, this process here and this diffusive process together, that you would also have something that's stable, no? However, Turing demonstrated that if the repression force diffuses at a faster rate than the activation, then a non-random pattern of some sort will develop. And this is what he called diffusive instability. So the basic idea is the following. The activating chemical start a reaction, and at a specific point in space, no? And it begins diffusing, and that's what I'm attempting to show here. So you have at a particular point the activation, and it starts diffusing. It begins its diffusion away from that point immediately, and then the repressive chemical is eventually produced by that reaction and start diffusing at a faster rate and cancel the effect of the activator, no? Because it's diffusing at a faster rate, a rate that is greater than the rate of diffusion of the activator, then, you know, it basically cancel it at some point in time. And then this basically result in spots. So you have a pattern that gets formed because this other chemical is diffusing at a faster rate. The inhibitor, no, is diffusing at a faster rate. You have the activation, and then the inhibitor catches up, and then you have the activation again, etc., etc. And so this figure here comes from Turing's original paper, and you know, this is what's generated in his with his equations. And so essentially this result in spots like the spots of the leopard, or stripes like the stripes in a jaguar, no? And so it's a really interesting way to look at pattern formation, no? And it's this combination of this diffusive instability that Turing talked about. This, this it can form these patterns, but it can form all sorts of patterns. I'm sure you've seen pictures like these of all sorts of pattern, spatial pattern formation that result from this process of, or these diffuse instabilities. We developed a cellular automata model that incorporate these basic processes. And here we have the ant clusters, no? And the budding process of the Azteca ant nest is the equivalent to the Turing activator. So the ants are expanding, no? That's with the budding process, the ants are expanding, that's the activator. And the phorids are attacking the ants and in a density dependent fashion, and that's the repressor. So we also have demonstrated that the phorids attack rate is density dependent. That is, that the higher the number of ant nests within a cluster, the higher the attack rate of the phorids. No, and that's what then it leads to this pattern formation of clusters. Here is the data from the field and the outcome from the simulation. So you can see in the left is the data from the field, on the right you see the simulations that came from this cellular automata model. The model predicts that the model basically predicts the number of Azteca nests and the cluster distribution to a relatively high level. Furthermore, we found that calculating the frequency of cluster size for the field data and the outcome of the model produces a very similar power function or power relation as would be suggested if the system is a self-organized system. Self-organized systems have this characteristic of a power distribution, no? Or a power function. And we did get that with both the model and the data. Now, it turns out that something similar to the Turing process happens in Hoffaker classical predator-prey experiments with the oranges. I don't know how many of you have read this classic paper, but in ecology is the paper that most graduate students probably have read. At least your faculty advisors have probably read it. But in this paper, Hoffaker was doing experiments with orange mites and its predatory mite, no? Except that in this case, the repressive component, that it's the predator-prey system, no, alone is unstable. So just having the predator-prey system alone in individual oranges, no, in each one of these oranges, there was a small population of the prey and the predator. And if you have them along, essentially what you have is a very unstable system where the prey increases, then the predator increases, and that leads to the collapse of the prey, and then both oops, sorry, let me go back here. Both of them go extinct, no? Along the system is unstable, but what Hoffaker discovered is that by adding the activator, in this case, the activator is the migration from orange to orange. So allowing these organisms to migrate from orange to orange, then you have a stable system. The system stabilizes, at least for a little while, no? And here you can see, these 3 cycles where the predator, you have the predator, the prey, the prey-predator system oscillating. And this is when the oranges were connected with each other. But one component that has not been explored that much in Hoffaker experiment is the spatial pattern that emerged in this system. So you probably have seen this. This is a classical, oscillatory pattern, but you probably have not seen the spatial pattern that was created. And here you can see it. This is from Hoffaker paper. And there is a special pattern that is generated in these oranges with the predator and the prey. And you can see it here. Now, this is something that has been developed theoretically by David Alonso in Spain and his colleagues. Here you can see the emergence of the spatial pattern for the prey at different points in time in a spatially explicit predator-prey model that he developed based on this the results from Hoffaker. And going back to Hoffaker classical experiment, we can zoom in one of the mite predator prey cycles. This is one of the cycle here. And so let's zoom in into that cycle and look at what happened with the prey and examine that spatial dynamics. And for example, here, you see that you have, a bunch of, this is the location of the oranges, and the densities are of the organisms. The shade of the square represent the density of the organism. So very dark means more, etc. And so you can see here that you have like a cluster here, small cluster, and that cluster grows, and then it grows even more, and then it's split into like two smaller clusters, and then one of the cluster disappear and one stay and then began growing. And if you choose this other area here, there's another cluster here, small cluster, that clusters grow, and then from here, and then it becomes even bigger, and then eventually it's split, and then eventually one of the smaller cluster disappear and the other one stay. No? So you have this very dynamic process that's going on, very similar to what we found with the Azteca nest in our 45-hectare plot. You can see something very similar happening with the Azteca nest. And you can see, for example, here, you can pick any part of this map, and you see the same kind of dynamic where here now we have in 2006, we have this very dense cluster here. And then in 2011, that cluster is getting a little bit smaller, but then you have the appearance of a cluster here. And then in 2016, that cluster completely disappeared, and then this cluster is beginning to grow. So it's a very similar dynamic. The system is very dynamic. Okay, so that's one of the vignettes. Now that we have established that there is a spatial pattern caused by the ants, let me move to the second vignette, which is about the beetle predator and critical transitions. Now remember that the ants are tending the green coffee scale, no? So here we have, this is a coffee plant with a high density of the scale insects, and you see the ants are foraging all over this plant, no? The ants nest on the shade trees, but they forage on the coffee tree. So you have the nest of the ant is on the shade tree, and then they forage on the coffee plants that are close to the shade tree where they're nesting. Because the scales are tightly associated with the ants, the density of the scales are high in the areas where the ant clusters are located. So, this is the entire farm. There's coffee all over here, but you find scale insects only in these areas that have very high density of ants, or only where the ants are located. Actually, I shouldn't say only where there are high densities of ants. High densities of scale only where the ants are located. You can find scales all over the farm, but they are at very, very low densities. Now, the scales have several natural enemies in the system, but the most important one that is particularly effective is this Coccinelli beetle called Asia orbigera. This is the beetle here, and this is the larvae of the beetle. It's really cute Coccinelli beetle. So both the larvae and the adult are voracious predators of the scale. To quantify the ant protection of the scales, we conducted an experiment. This is the work of one of my students, Shinsuke Uno, and he basically have branches on the coffee bushes and isolated put tangled food to prevent the ants from going to certain branches that have scales, and he allowed ants to go to branches that also have scale. And you can see how the dramatic reduction in this, I'm sorry, I forgot to put the label here, but these are numbers of scales. So the dramatic reduction in the numbers of scales in the areas where the ants were not allowed to allow access, no? And so basically in 3 weeks, the scales were gone. This predator basically ate the scales. So the ants are very effective at protecting the scale insects. And the ants are fairly aggressive when they are tending scales and attack any insect that come close to the scales. And the beetle adults are no exception. No, they attack the beetles when the beetles get close to the scale, and usually the beetle is able to fly away because they can fly. So as soon as they get approached by an ant, they fly away or drop to the ground or something like that. But sometimes, every once in a while, like you see here, the beetles are able to grab the beetle. I mean, the ants are able to grab the beetle and kill it. But most of the time they do fly away. Now, so the beetle eats the scales, but the ants interfere with the ability of the beetle to prey on the scale. No? And so here I'm indicating the beetle eating the scale with this arrow positive and the ball is negative. So this is a direct predator-prey interaction. And then I'm illustrating the interference, which is a trait-mediated indirect effect or a higher order effect, is the ants interfering with that interaction. So it's a negative effect, no, that the ants have on that interaction. Since the system is spatially explicit, because I already showed you that the ants are in clusters, and the ants form, these clusters within the coffee farm, the ant patches represent refuges for the scale insects. No, that's where the ants are, and the scales are being protected by the ants. But let's zoom in to the level of a shade tree and the coffee bushes that are surrounding it. Very close, no? The ant nest on the shade trees, or on one shade tree, tend the scales on the adjacent coffee plants, as I mentioned before. And the scales produce crawlers. You see these little scales here, these are crawlers, and they can move around, and the crawlers disperse to other coffee plants. But in order to survive, they need to be found by the ants, no? And the ants then start tending them and protecting them. So you have, the adult scales producing crawlers, the crawlers disperse, but then they need ants to protect them, because otherwise then the predators will eat them. Now, the question is, how far will the ants go to tend the scales? Well, it depends on how many scales there are. So very close to the nest, because that's where the high concentration of scales are, and they are dispersing farther away. So close to the nest, ants recruit other nest mates because the scales are at high density. However, far from the nest, you might get a crawler that disperse farther away. And here you can see, this single individual scale that fortunately had not been found by a predator or a parasitoid, no? But that's rare, but they can survive. Every once in a while they do survive, but they are not protected by ants, because it's not worth for the ants to go there to tend one single individual, especially if it's far away, no? It's not worth for the ant. So the success of the scales depend on the protection from the ants, and ants tend scale only when they reach a critical density. So this we're calling the Ali effect, no? The scales need to have some sort of critical density for the ants to, be able to, or be worth for them to invest the energy to go all the way there and start tending them, etc. Below that critical density, it's not worth for the ants to tend them. So here we have the exponential growth equation in a discrete form. And here is the equation with some density dependence. Some of you are familiar with these equations. And I just added the Ali effect. We introduced the Ali effect as basically n-crit. So n-crit is the critical density of the scales that is needed for the ants to start tending the scales. Now, constructing an iterative map of this equation, we have this function here. And this is an n of t against n of t plus 1. No, the population of the scale at time 1 versus the population of the scale at time t plus 1. And we put a 45-degree line here just to look at the dynamics, and to help us visualize the dynamics. If you start over there, then you move along here, 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 and eventually end up there. And that's an equilibrium point, no? We have another equilibrium point here, which is at 0. So if the population of scales never reach this critical density, it will go down to 0. If it reached that critical density, then eventually it will increase to this higher level equilibrium point. How can the Ali effect be translated into distance from the nest or distance from the refuge? So we propose that this is what is happening, okay? When basically when the scales are dispersing toward the refuge, when they are dispersing from outside of a refuge where the ants are toward the refuge, you have very few scales, and it's not worth for the ants, basically. It's not worth for the ants to tend those scales unless they get very, very close to where the ant nest is, no? But until they get very close to where the ant nest is located, then it's not worth for the ants to be tending them. And the scales have a hard time surviving attacks by natural enemies. So when the scales are dispersing from the refuge to outside of the refuge, the ant keep moving farther and farther away of the nest, because there are many more scales that are dispersing, and so it's worth for the ants to keep moving a little bit farther. Obviously, if it's too far, then they won't go that far. But there is a tendency for the ants to be able to, to move outside of the refuge and limit the mortality of the scales by the natural enemies. So let's look at what happened then as we change n-crit, the density, the critical density of the scales. So remember the equation here, we added this, which is the Ali effect, that represent that density, that critical density. So if we have a very high n-crit, our function is going to look something like this, and that means that there's that the only equilibrium point is going to be right here, no? If we increase the n-crit, if you decrease the n-crit, getting closer to 0 here, then still you don't get to intersect the line, and you still have this equilibrium point at 0. Now, if you increase it, if you decrease it even further to 0 in this particular case, or very close to 0, then you have basically one equilibrium point, which is this point here at high level of the scale. You decrease it even further, you decrease it even further, you still have that equilibrium point basically at the very high level of the scale insect. Now, and in the intermediate between these low levels, or I'm sorry, between these high levels of the encrete and these low levels of the encrete, you have the situation that I showed you before, where you have two stable equilibrium points and one unstable equilibrium point, no? And here we have a summary graph of the equilibrium points as we see the changes in encrete. So what I'm doing here is I'm going to be plotting the equilibrium points with different levels of the critical density, no? And here you see that here you have the high densities at low critical, at the low critical points or low encrete, you have the high density of the scale. At the high encrete, you have the low density of the scales. And at this intermediate level, you have both high and low and basically an unstable equilibrium point. In other words, as we move from very low encrete to a higher encrete, we stay at very high scale density until we reach a critical transition point where basically the density drops to 0. And then so that's a critical transition point. And then if we move basically from these very high levels of encrete toward the low levels of encrete, we move in this direction until we reach another critical transition point, but this time is a critical transition that goes from 0 to high levels, but that's also a critical transition point. And then this produces a region of instability or what is called a hysteresis or a hysteretic zone, no? So in this area, you're going to have high levels and you're going to have low levels as well. So this blue area represents dispersion away from the refuge, and the red area represents dispersion toward the refuge, no? And you have then the hysteretic zone in between. So what we did then, after doing, that, just, playing around with the model, we sampled the ants on coffee plants at various distances from the tree where the ants where the ant was nesting, and we get this graph here. And as you can see, this is distance from the nest, and this is the number of active ants per minute. And you can see that there's a lot of ant activity all the way to about 4 meters, between 3 and 4 meters from the tree where the ants are nesting. These measurements are all made on coffee plants, no? And you can see that there's a lot of ant activity, and then it drops to almost 0 after the 4 meters. Then sampling the scales on those coffee plants, basically what we get is this. And you can see a critical transition with a hysteresis zone. Very close to the nest, all the coffee plants have very close to the nest, like here, all the coffee plants have very high densities of scale. Far away, after the 4 meters and even farther, you have very low densities of the scales. Now, in between, in that hysteretic zone, you have a lot of plants that have high levels, a lot of plants that have very low levels, and some in between. No? So basically, we found this critical transition zone and a hysteretic zone in the scale insects as you move away from the nest site of the ants. Okay, So now that we have established that there is a spatial pattern caused by the ants that affect the scale spatial pattern distribution, and that there is a critical transition with distance from the tree where the ants are nesting, let's then examine another important component of the system, and that is a fungal pathogen that attack the scales and that we think may cause chaos and another histolytic zone very close to the tree where the ants are nesting. So this is Lecanicilium lecani, is a fungus that attack the scale insects and that cause episodic. When the scales get at a very, very high density, as you can see in this picture, every single one of these scale is dead, has been attacked by Lecanicilium, and they're dead. And this can happen actually fairly fast. This fungus spread fairly fast among the population of the scales. So the fungus tends to form local episodic on the scales when the scale is at a very high density, but rarely occurs if the scales are at very low population densities. Now, remember that the scale population is high when it is protected by the ants. So the ants are facilitating that interaction. And that's why I put this, a kind of higher order interaction here with the arrow, which means positive. So the ants are facilitating that interaction. Also remember that the scales are eaten by the beetle. No, the beetle is the predator of the scale. But close to the nest, to the ant nest, the beetles are harassed by the ants. So that's another higher order interaction, but this time is a negative higher order interaction. So the question is, what happens in this zone? No, what happened in this area here? No, in this area that is very close to the nest, to the ant nest, where you can have this episodic of, the fungus. We should expect another critical transition with another hysteresis very close to the nest. Now, so in this zone here, we should expect, another critical transition and another hysteric zone. Now, remember that the iterative map that I showed you before, this iterative map here, what happens, we ask what happened if R increases, R meaning the rate of growth of the scale population. If R increases here in this equation, as it does when the scales are very close to the ant nest, then we have something that looks like this. So the shape of that curve, that function changes, with the increase in R. And that can lead to chaos. Essentially, that in these areas where you have these very, very high populations, that will lead to a chaotic behavior. Just with that simple equation, adding the encrete and adding, basically with a high rate of growth of the population of the scale. So with changes in encrete and an increase in R, we get something like this. And translating that to distance from the refuge, so we get this chaotic zone here. I'm expanding a little bit the area, of that is close. Well, basically the encrete, this is not spatial, but this is the encrete. And you can see here, but if we look at that in terms of distance from the nest, and now here I have expanded this area a little bit so that we can see what happened. This is our original critical transition zone that I mentioned before, that comes from the natural enemies, the dispersal into the refuge and out of the refuge. And now I have expanded the area from the nest into this critical transition zone and this hysteretic zone so that you can see that other area of chaos, and that is basically something that we're exploring now. We're in the process of gathering data. So the models say that this is what should happen. Now, we don't have the data yet to prove that this happens. No, we're in the process. We were in the process of gathering the data when the pandemic hit, so we had not been able to continue doing the field work there, and so stay in tune for next time. But next time when we go to Mexico, we're hoping to basically zoom in that region and try to figure out whether we are able to gather some evidence that show that indeed there is another critical transition and there is a chaotic zone there. Another wrinkle on all of this is that the fungus has a strong seasonal component. Actually, the whole system has a seasonal component and needs to be explored. In the dry season, everything doesn't shut down, but the populations go down a little bit, and then they increase in the dry season. So that would be interesting also to look at. Now, finally, I should also mention that the ants also provide a refuge for the beetle larvae, and I didn't talk about this at all, which is the beetle larvae is protected from the ants with these waxy filaments. So the ants cannot prey, cannot kill the beetle larvae. And that creates another whole set of interactions there that I don't have time to talk about. The ant clusters represent sources of adult, in a nutshell, the ant clusters also represent sources of the adult beetles that are controlling the scale insects in the rest of the farm. So in addition to being a refuge for the scale insects, there are a refuge for the predator of the scale insects. And that allows for the control of the scale insect in the entire farm, even in areas where there are no ants. No Azteca ants, I should say. Now, I know that it's taking me quite a long time, but I want to take just a few minutes to talk a little bit about the ecological game that we develop. And so let me just say that what I presented so far is a very small fraction of the web of interactions that are related to the control of 4 of the main pests in coffee. No, and all this network of interaction has a keystone species, which is the Azteca ant that I talked about before, and so what I show you today were these interactions only, including Azteca, the insect, the scale insect, the white hello fungus, Lecanicilium lecani, and Azaya orbigera, the beetle. No. But then there are all these other components that are really interesting as well. A lot of interactions, direct and indirect interactions happening there. But I mean, it's difficult to communicate this to farmers, and even more difficult to tell them that this Azteca ant is actually very important for the control of all these pests, because the farmers, when you talk about ants and the importance of ants in the agroecosystem, they look you with a very suspect eye. They don't like ants because the ants, some of them sting, and Azteca bite actually. It's kind of annoying, a little bit annoying. And they can see that the Aztecas are tending the scales. No, so they are concerned about the Aztecas protecting the scale insects. So they look at ants like the enemy, and the enemy that needs to be vanquished. However, getting rid of the ants might imply the collapse of the biological control in the rest of the farm. So how do you communicate this to the farmers? And taking part of these interactions, and I didn't talk specifically about this. I talk about the ants tending the scale and the beetle eating the scale. I didn't mention how the forage fits into all those interactions, and the parasitic wasp that attack the beetle, etc. But we decided to develop a game that include at least this core of interactions here. And so the main idea is to try to communicate the complexity of nature to the farmers. And so we have this game. There are 2 players. It's called Azteca chess. There are 2 players. The Azteca ant and the killer wasp is played by 1 player, and then the adult and the larvae of the beetle is another player. The other components, the headhunting fly, which is the phorid fly, the honeydew, and the scale insects are kind of autonomous component of the system. They're not moved by the they're just stationary, and they're not moved by the players. The Azteca, the phorid fly, move autonomously. Every round there's a movement of the fly, but the players don't move it. So here's the board of Azteca chess. This is the position of the phorid fly. So at every turn, the phorid move from here to position 2, and then the next round from 2 to 3, the next round from 3 to 1, etc., etc. And this is how the board looks like at the beginning of the game. These are all the components. Each one of them, these are the rules of the game. And it incorporates the direct interactions, predator-prey interaction, and the reproduction of the organism. And it includes also the trait-mediated interactions, the higher level interactions. Like, for example, you have here the phorid fly. When the phorid fly is right next to the ant, then the ant needs to leave the table. No, so the ant disappears. It's kicked out, and you lose your ant if it comes, to a cell that is next to the phorid fly, etc., etc. No, so we have played this with coffee farmers, primarily in Mexico, but also in Puerto Rico and here in the United States. Here you can see some of the farmers. They have a lot of fun playing the game. At the beginning, they're a little bit intimidated with the rules of the game and all that, but eventually, very quickly, they catch up. And even the kids play the game, and actually the kids are much better at it than the adults. But yeah, we have a lot of fun playing the game. And I want to show this picture because this was one of the coffee farmer that played the game in one of the tournaments that we had in one of the communities. And 2 weeks later, he happened to be a wood carver also. And 2 weeks later, he showed up in the office of my colleague in Mexico, Luis Garcia, who was kind of the brain behind the game, with this game that he carved himself based on the pictures of we left a copy of the game, but we play it with these cardboards and just little cardboard tokens, no? And based on the pictures, basically he carved this game himself and painted it and all that. So now we order games from him whenever we go to a place to play the game. We leave one of these games with the community. And then finally, I would say that I was giving a talk about this system, and there was a student that was an engineering student from India, and she needed to do a project, and she got fascinated by this game, and she said, oh, can I try to do this game for an application for a smartphone? And I said, yeah, sure, go ahead. And so she developed this game, Azteca phone app, for the phone. Her name is Kirti Talam, and the game is not it's not active yet, but she's in the final phases of doing this app for the smartphone. And so with that, I'm going to finish, and I'm sorry that I went a little bit over. I wanted people to have time to ask questions, but if you have any question, I'll be happy to answer.
Great. Thank you so much, Dr. Perfecto. Awesome talk. Really enjoyed it.
Thanks.
We do have some time for questions. I know that some people might have to leave, which is